신기하게 어떻게 하냐면 이런 식으로 하고 국내 가족 이렇게 해서 근데 여기 걔네들 CC도 안 했어.
이거 아무도 안 했네.
의견을 물어본 것도 아니고 난 내가 처음에 몇 개 했으면 좋겠다 하니까 이렇게 개수 올리자고 해서 다 올려주고
Hello? Hello, yeah, can anybody hear me? This is Kelly at Logos Biosystems.
Oh, yes, Kelly. Hello. I'm Azalea.
Oh, I cannot hear you. Wait a minute.
Oh, okay. Wait a minute. Oscar, can you please turn the music off first?
Yes, sorry.
Okay, I can hear. Okay, can anybody hear me? This is Kelly at Logos Biosystems.
Oh, okay. Hello, Kelly.
Hey,
I'm
hello.
Azalea Putri.
All right.
Application specialist at Plasma Biotech Global.
Hey,
It's really
hello.
nice to see you.
Yeah,
Hello.
me too.
Okay, currently we're still waiting for the attendance for the
Okay.
webinar.
Okay.
So we will start probably around 1:30
Okay.
as from the poster that we already made, right?
Okay.
Okay.
Okay, what to pretend I will turn off my camera. I'm shy.
Okay.
Okay, Kelly. It's okay to turn on the video.
Okay, while waiting, I think it would be better, Oscar, you can turn the music on again, or you have something to say as a moderator.
Halo, selamat siang. Apakah suara saya terdengar? Halo.
Dengar, Pak.
Dengar, Pak.
Siap. Selamat datang Bapak Ibu, rekan-rekan seluruh peserta webinar kita siang hari ini. Kami mohon untuk selama webinar ini berlangsung untuk memute audio masing-masing. Tapi kami minta untuk seluruh peserta boleh menyalakan videonya. Ya, jadi supaya kita bisa saling melihat ya, saling sapa. Jadi bisa dinyalakan videonya, tapi untuk audio mohon untuk di-mute selama berlangsungnya webinar kita siang hari ini. Selamat datang untuk semua yang baru bergabung, baik itu dari Zoom meeting maupun yang bergabung melalui live YouTube kami. Halo, selamat siang semuanya.
Selamat siang, Pak.
Ya, sekali lagi selamat siang, selamat bergabung di webinar kita. Untuk Bapak Ibu dan seluruh peserta yang bergabung pada webinar kita siang hari ini, dimohon untuk mematikan audionya selama berlangsungnya acara webinar dan dimohon untuk tetap menyalakan videonya agar mungkin kita di sini bisa saling menyapa ya. Ya, kita masih menunggu beberapa peserta lagi yang mungkin belum masih terkendala untuk bergabung di Zoom meeting ini. Tapi beberapa juga mungkin sudah bergabung melalui live YouTube kami. Selamat datang, selamat siang untuk Bapak Ibu, rekan-rekan seluruh peserta pada webinar kita siang hari ini. Nah, untuk Bapak Ibu dan rekan-rekan yang bergabung melalui Zoom meeting, dimohon untuk menonaktifkan atau memute audionya selama berlangsungnya webinar, dan mungkin bisa dipastikan untuk videonya agar tetap menyala supaya kita di sini bisa saling lihat dan saling menyapa. Jadi video boleh tetap dinyalakan, namun audio mohon untuk dimatikan atau di mute selama berlangsungnya acara. Terima kasih. Baik, masih banyak nih yang baru bergabung kembali di Zoom meeting, dan di live YouTube juga sepertinya semakin bertambah yang hadir. Oke Bapak Ibu, rekan-rekan seluruh peserta mungkin yang bergabung melalui Zoom meeting ini boleh dinyalakan videonya, dan untuk audio, tapi untuk audio selama berlangsungnya acara mohon untuk dimatikan. Ya, terima kasih. Ya, kembali saya ucapkan selamat datang untuk Bapak, Ibu, dan rekan-rekan seluruh peserta yang baru bergabung, baik melalui Zoom meeting maupun melalui live YouTube kami. Selamat siang, selamat datang semuanya. Untuk yang bergabung melalui Zoom meeting, kami mohon untuk menonaktifkan audionya selama berlangsungnya webinar kita, tapi kami juga mohon untuk boleh menyalakan videonya. Ya, jadi untuk yang videonya belum nyala mungkin boleh dinyalakan. Ya, ada beberapa yang nyala dan masih ada beberapa yang masih belum dinyalakan videonya. Boleh kita mungkin bisa saling lihat live-nya. Bapak Ibu semua di sini memberikan senyumnya kepada yang lainnya supaya spiritnya itu sampai ya seluruh peserta juga gitu. Bapak Ibu rekan-rekan yang sudah bergabung baik melalui Zoom meeting dan YouTube video, kami mohon kesabarannya karena di sini masih terus banyak yang baru masuk lagi, baru join lagi baik itu di Zoom meeting dan live YouTube. Jadi kami mohon untuk menunggu sebentar lagi sebelum dimulai.
Ya endan ora tumbas. Ora, jero kan tak takoni endan gak sadar apa gak.
Sekali lagi saya mengingatkan untuk seluruh peserta webinar pada siang hari ini, kami mohon untuk boleh menyalakan videonya. Selama berlangsungnya acara, kami mohon untuk tetap menyalakan videonya. Namun untuk audio mohon dinonaktifkan atau di-mute selama berlangsungnya webinar kita. Saya ucapkan selamat datang, selamat siang untuk seluruh peserta yang baru bergabung, baik melalui Zoom meeting maupun melalui live YouTube kami. Semoga semuanya tetap dalam keadaan baik di tengah-tengah kondisi pandemi seperti ini, tetap jaga kesehatan, tetap jaga protokol yang dianjurkan oleh pemerintah agar kita semua tetap sehat dan dapat menjalankan aktivitas kita seperti biasa. Nah, siang hari ini kita akan mengadakan webinar. Nah, webinar ini pastinya akan berlangsung sangat menarik. Jadi mohon untuk update baik itu di Zoom Meeting maupun di YouTube kami. Dan nanti kami juga akan, sedikit bocoran, nanti kami akan memberikan promo atau penawaran spesial untuk seluruh peserta yang hadir pada siang hari ini. Nah, jadi tetap stay di tempatnya masing-masing selama berlangsungnya webinar kita siang hari ini. Nah, untuk Bapak, Ibu, dan rekan-rekan peserta yang hadir melalui Zoom Meeting, yang bergabung melalui Zoom Meeting, kami persilakan untuk menghidupkan, menyalakan videonya. Namun untuk audionya mohon di mute atau dinonaktifkan selama berlangsungnya webinar. Masih menunggu beberapa peserta lagi yang baru bergabung, yang masih akan bergabung, karena ini sepertinya masih terus bertambah. Jadi harap bersabar untuk Bapak, Ibu, dan rekan-rekan seluruh peserta yang sudah bergabung. Terima kasih. Ini pesertanya, mungkin beberapa menit lagi kita akan mulai. Oke, apakah masih ada yang bertambah lagi? Oh ya. Baiklah, tapi mungkin karena waktu sudah menunjukkan pukul 1.30 ya. Baik. Kita langsung mulai saja webinar kita siang ini. Selamat siang, salam sejahtera untuk kita semua, Om Swastiastu. Selamat datang Bapak Ibu dan rekan-rekan seluruh peserta webinar kita pada siang hari ini yang bertema How Cell Counter and Cell Imaging System Help Your Research and UC. Nah, sebelum itu perkenalkan terlebih dahulu, saya Oscar dari Plasma Unitek Global. Sebetulnya saya hari ini tidak sendiri, saya nanti akan dibantu dan ditemani oleh rekan saya yaitu
Halo, selamat siang. Perkenalkan saya Azalia Putri, Application Specialist dari PT Plasma Unitek Global.
Ya, jadi saya dan Kak Azel yang akan memandu jalannya webinar kita pada siang hari ini. Nah, sebelum kita memulai webinar siang hari ini, mungkin saya kembali mengingatkan kepada seluruh peserta, Bapak Ibu rekan-rekan seluruh peserta yang hadir, khusus untuk yang bergabung melalui Zoom meeting untuk menonaktifkan audionya atau mengganti ke mode mute dan kami persilakan untuk tetap menyalakan atau turn on videonya selama berlangsungnya acara webinar kita siang hari ini. Dan apabila nanti selama webinar materinya disampaikan, Bapak Ibu dan rekan-rekan seluruh peserta boleh memberi pertanyaan melalui kolom chat yang tersedia di Zoom meeting maupun kolom komentar yang bergabung melalui live YouTube kami. Nah, jadi untuk yang bergabung melalui live YouTube, selamat siang semuanya. Boleh diramaikan kolom komentarnya. Nanti saat berlangsungnya webinar boleh memberikan pertanyaan sebanyak-banyaknya. Nanti akan kami coba untuk membantu menjawab terkait kebutuhan Bapak Ibu dan rekan-rekan seluruh peserta. Nah, webinar kita siang hari ini diadakan oleh PT Plasma Unitek Global. Untuk itu saya boleh bacakan sedikit mengenai company profile PT Plasma Unitek Global. Jadi PT Plasma Unitek Global ini berdiri sejak tahun 2017 dan tim yang membentuk PT Plasma Unitek Global ini sudah lebih dari 20 tahun berpengalaman di bidang sales dan marketing produk-produk life science secara umum, dan kami Plasma Unitek Global membawa visi dan
Putus-putus.
Customer kami satu harapan dan
Putus suaranya.
ya maaf mungkin suara saya putus-putus. Ya, jadi kami bukan hanya distributor untuk menyediakan produk-produk, tapi kami juga menawarkan jasa dan juga kami akan sering mengadakan sharing session seperti webinar kita siang hari ini. Nah, untuk PT Plasma Unitech Global ini kami berlokasi di Rukan Grand Orchard Block GOS untuk menghubungi tim PT Plasma Unitech Global. Ya, jadi sebagaimana yang dapat Bapak Ibu dan di layar mendukung kegiatan plasma 2. Baik Bapak Ibu, maaf mungkin ada sedikit gangguan di koneksi saya. Kembali saya akan share screen. Nah, oke. Sudah aman lagi ya. Baik. Nah, di PT Plasma Unitech Global ini kami menawarkan beberapa produk dari beberapa brand seperti reagen Jenny, Mini Nes, Denatrasia, Anbiotek, Gloveon, SRL, Logos Biosystem, Leptron maupun Labdes. Nah, untuk DNA Tracia ini sendiri merupakan kit DNA tracer yang biasa pengaplikasiannya untuk bidang bioteknologi. Yang digunakan untuknya teknologi yang dapat memudahkan banyak orang untuk dapat menggunakan teknologi DNA atau yang biasa dapat digunakan di mana PCR itu ini brand Nes. Nah, ini menawarkan jenis plasticware, salt solutions, kebutuhan reagen-reagen atau bahan-bahan kimia secara umum dalam berbagai kegiatan itu kami menawarkan SRL atau Cisco, ini merupakan brand Indodigeni dan Elisa. Terakhir, untuk peralatan-peralatan umum, life science secara umum, hampir 100 dari produk tersebut, kami dapat mendukung beberapa kegiatan seperti sel bio, NB process, genomics, general lab, applied science, maupun proteomics, seperti itu. Area market share kami sudah 70% berpusat di Pulau Jawa, dan kami juga sudah mengembangkan area market. Di Universitas Sriwijaya, lalu institusi penelitian seperti pertanian, salah satunya BB Biogen, lalu pusat pendidikan, Pusat Penelitian Indonesia, LIPI, dan juga Kementerian Kesehatan Republik Indonesia. Karena beberapa institusi swasta juga atau industri-industri yang sudah bekerjasama dengan kami seperti Danone, Garuda Food, Otsuka, dan beberapa perusahaan farmasi seperti Kalbe, Dexa, Bio Farma, Capri Farmindo, dan ini Sinarmas untuk industri agribisnis, sektor agribisnis. Nah, jadi dari beberapa profil customer kami, diantaranya bergerak di farmasi, stem cell, maupun pengembangan vaksin. Nah, kami juga bisa support untuk beberapa rumah sakit seperti SCM, Rumah Sakit Darmais, dan lain sebagainya. Baik Bapak Ibu, dari yang sudah saya terangkan tadi, kami mengangkat tema untuk webinar kita siang hari ini dari brand Logos Biosystem. Jadi ini salah satu produk unggulan kami karena ini merupakan teknologi automated cell counter, cell imaging, tissue clearing, and 3D imaging. Mungkin selanjutnya akan dibantu oleh Kak Azel untuk memperkenalkan pembicara kita yang jauh-jauh nih dari Korea Selatan. Nah, jadi dipersilakan kepada Kak Azel untuk mempersilakan speaker di hari ini.
Halo. Ya, terima kasih banyak Oscar atas presentasinya. Ya, mungkin selanjutnya saya akan memperkenalkan narasumber kita pada hari ini, yaitu Kelly Kim sebagai produk spesialis dari Logos Biosystem Korea. Hello everyone. Now I introduce the speaker for today's webinar. Oke, mungkin untuk reminder lagi, untuk mengingatkan kembali, bagi para peserta yang memiliki pertanyaan, bisa langsung mengajukan pertanyaannya pada kolom komentar. pada kolom komentar dan semua pertanyaan akan dijawab pada sesi question and answer secara live dan sisanya akan kami follow up melalui email. Also, following the webinar, please add any question that you might have to chat, and any question will be answered during the live question and will be followed up later by email. Saya sudah terdengar? Hello, Miss Kelly. Can you hear me?
Yes.
Can you hear me clearly, or?
Yeah, this is Kelly. I can hear you very clearly.
Oh, okay. Okay, great.
Okay.
I already introduced you as a speaker for today's webinars.
Thank you.
And how are you today, actually?
Really good. This is Kelly Kim, everybody, and from Logos Biosystems. And today it is good. And thank you for introducing me to everybody. Yeah, that everything is going well. So that, how about in Indonesia?
Okay, in Indonesia also we are great. Also, we still fight with coronavirus.
Okay.
And yeah, could you tell us a little bit about this webinar? I mean, about the material that you want to present?
Okay. You mean that to everybody here?
Yeah, just a little bit.
Okay, then today we will talk about introduce our cell counter. So you will learn that how you can choose which cell counter you need to use for your research. And also we will introduce about our Selena fluorescence imaging system. It is actually already widely used in worldwide, the conventional microscopy, but I'll introduce why we need a digital imaging system for your research, especially to improve your current research status. It will be helpful for you. So it will, and for the cell counter, our European director Karin Labor will introduce about cell counter, and I will introduce about the Selena imaging system today.
Okay. Okay, thank you very much, Kelly, for a little bit information for today's webinar. So probably from now on, we will present the presentation about the cell counter first, right?
Okay.
Okay. Okay, please, Oscar, can you help us?
My name is Karin Labour. I'm Europe Managing Director at Align Genetics. Welcome to this How to Choose a Cell Counter webinar. The webinar will last about 30 minutes. And during the presentation, you can ask questions using the question and answer box in Zoom. And at the end of the presentation, I will answer questions, some of the questions, depending on the number of questions. If we can't answer all the questions today, we will answer the questions by personal emails. So just a few words about Align Genetics. Align Genetics was founded in 2008. This is a mid-sized company with a large network of partners worldwide, with a deep focus on innovation. We have two brands. GenScale, which is an in vitro diagnostic point-of-care solution brand, and the Logos Biosystem, which is already a leader in imaging solutions for life science, including digital microscopes, tissue clearing solutions, and cell counters. And today we will talk about cell counters. So cell counting is a necessary, but most of the time a tedious step for in vitro cell culture. But this is really important when you want to monitor the health of your cells, you want to check the proliferation rate, you want to assess metabolization, transformation, you want to check the transfection or infection, or any seeding, anytime you want to seed cells for subsequent experiments, especially for cell-based assays. So it's critical that your cells are counted accurately with reproducible methods, especially when you want to quantify measurements of cellular responses. And among the challenges when we talk about cell counting, well, the first challenge is actually the cells themselves. You have a huge variety of cells available that you may want to count. Can be a bacteria. Bacteria, they have a very small size, very small genome size, can be like 1,000 times smaller than mammalian cells genome size. Or you can have budding cells like yeast, or even if you're working with mammalian cells, you don't always work with cell lines. You can face the presence of debris or contaminants like red blood cells if you're working with PBMC leukocytes. Also, the cells can have different, in the same sample, you can have different sizes, and that's the case for MSC cells, for example, or you can have clusters of different shapes. But those are standard cells, and maybe you're interested in counting other kind of cells like pollen, protoplast, spores, sperm, ovocytes, microalgae, parasites, etc. So the diversity of cells is the first challenge to face. And you know what? 5 years ago, still 70% of the researchers are considered that the hemocytometer was still the gold standard for cell counting. So we all know what is a hemocytometer. So you have the mounting support, you put your cover slip, you load your cells, and then you're going to count with a grid as a reference. This is a very old method. It was created by Mr. Louis-Charles Malasset, a French guy, more than a century ago. And this is still used by many, many labs. And it has lots of frequent issues, lots of issues. The first issue is, what is the perception of a cell? If you ask 3 different people to look at messy samples, not the same cells will be counted. I'm not talking about pipetting errors, dilution, volumes, or maybe sometimes, the pipettes are not even calibrated. Also, when you count under a microscope with an hemocytometer, it's tedious. It takes a lot of time. So you're not going to count lots of events, not lots of cells. And based on those low numbers, maybe you're going to define a concentration. So you can imagine the variability in the results. Another aspect is the difference is the inter-operator variability. So if you take people with little experience, you can see up to 100% differences in counts, which is not acceptable today for life science experiments. But even with highly experienced people, the minimum variability you can expect is 15%, which is outstanding. But still, lots of people are using it. And also the dye that you're going to use for the counts, and we will talk about the dyes. So of course, cell counters can solve manual counting issues, but choosing the counter, which is right for your needs, will require some precautions, some steps to be sure that you select the right instrument. And look, at Align Genetics, we already have 7 cell counters. 7. So we didn't develop 7 cell counters just for fun. It's really because you need different technologies depending on your cells. So today we will cover some of the aspects, I mean, this presentation will last 30 minutes, so I can't cover everything. But I will cover the aspects which seem the most important to me to make sure that you select the right counter. So let's start with the dyes. The most common dye and the most well-known dye is trypan blue. So trypan blue, as you know, it's an occlusion dye. It's a polar molecule, so it doesn't enter cells if the membrane is intact. And actually, I don't know if you knew it, but the origin when it was found, it's because it was killing trypanosomes, you know, in the sleeping sickness. This is an insect-borne parasitic infection of humans, and that's caused by Trypanosoma rosae. So the principle is very simple. When you mix your cells with the same volume of trypan blue at the right concentration, usually it's 0.4%, the alive cells won't let the dye enter, so they're going to be bright and not colored. And if the membranes are damaged, it's going to stain the cell, then it's considered as dead. This is interesting because obviously it's inexpensive, it's easy to use, you just mix the same volume of dye and cells. It's compatible with hemocytometer or image-based cell counters, and that's perfect for easy samples like cell lines. However, this method, this dye, doesn't allow to differentiate debris nor contaminants. A debris will be dark blue, so how do you differentiate a dead cell from a debris? No contaminants, red blood cells are dark red. Okay, so dark red versus dark blue, except if you use a high magnification, meaning you're going to spend even more time to count the cells, it's not relevant. So it's not accurate for complex samples. And another aspect is that this is a highly toxic compound. This is a mutagen, teratogen, cancerogen. Okay, so anytime you're working with trypan blue, be careful, use gloves. If you have some trypan blue on the surface, consider it as contaminated, decontaminate it. And luckily, we propose a safe alternative, the erythrosine B. So if you're working with trypan blue, maybe consider to switch to this safe dye. This dye is actually a food stain, and it's pink, it's not blue, it's pink, but it's working exactly in the same way. And that is safe. And we are the only one to propose it. So trypan blue is a common dye. Now there are other dyes like fluorescence dyes, nucleic acid dyes. The most common are DAPI, acridine orange, and propylium iodide. I selected to talk about acridine orange and propylium iodide because that's extremely relevant for cell counting. So if you look at the image on the right, this is a sample, a messy sample. You see that if we go to count it under a microscope, we would be in trouble because none of us would exactly count the same number of cells. But here, this is a PBMC sample from COVID patients. So if you add acridine orange is a cell permeable. So it means that it's not toxic for the cell, it will enter the cells and it will interact with DNA and RNA. If you add propylium iodide, this is a toxic compound and it doesn't penetrate the cells if the membrane is intact. And once the two dyes are in the same cell, there is an effect which is called fret that you probably know. So the acridine orange is quenched by the propylium iodide. So it will turn off the signal of the acridine orange. But let's have a look at those two cells. Okay, let's go a step back. So if I go back to the first image, you see that the first cell on the top, you would probably count it as a live cell or at least as a cell. When this one at the bottom, you're not sure whether this is a cell. But actually, when you look at the penetration of the acridine orange, you see that the bottom cell here actually has nucleic acids when this one doesn't have it. And propylium iodide doesn't enter any of those cells. So it means that it removes the subjectivity of counting with this kind of dyes. So in terms of advantages, acridine orange and propylium iodide are specific to nucleated cells, nuclei. It doesn't count debris nor erythrocyte. It's compatible with all kinds of cells except bacteria, because this is a specific situation for bacteria. But here you can count cell lines, but also more complex samples. However, you need to have the correct technology, the correct counter, fluorescent cell counter, to use it. And so one tip, it's not relevant if you're working with cells coming from chicken or fish, because for those species, red blood cells contain nucleic acids. So if you work on chicken or fish, I'm sorry, but there is no obvious solution at the moment. When it comes to yeast, we face another challenge, okay? And acridine orange can be used in combination with propidium iodide, but it's not super efficient because of the size of the cells and the limited emission of fluorescence. So one alternative is the combination of FDA, fluorescein diacetate, and propidium iodide. So propidium iodide works the same than for mammalian cells. FDA, it's a probe which can enter by diffusion inside the yeast, and the alive yeast will use their esterases to cut the FDA and to release fluorescein. So it's an indirect method which can be used to efficiently count yeast in combination with the propidium iodide. So those options are, so you see that there are different validated dyes. I didn't cover all the dyes which are available in the market because that would be too long, but those are the main and most relevant dyes. Okay, so now which technologies are able to detect those dyes and efficiently count your cells? So you have the hemocytometer in the microscope. We discussed it, it's a tedious method. Cytometry. You have the possibility to use cytometers. You're going to get much more information that's only the count, but this is highly expensive, time consuming, and maybe you don't have access to a cytometer in your lab. Third method is the Coulter method. This is an interesting method for particle counting, but far less for cell counting. This method is based on impedance measurements, so the changes in electrical conductance as the particles pass through a small orifice. When it comes to cell counting, the major pitfalls of that technology is the price. You can go up to €4 or $4 per count, which is far too expensive. The lack of control, you don't see your images, and it's not efficient when it comes to samples contaminated with debris or contaminants. So in the market, the image-based counters are the most common and affordable solution to count cells with those variability dyes. So if we say image-based counters, it's because it takes images, okay? It takes a picture of pictures. So the first technology for image-based will be the most simple one, the most direct one, so based on bright field. So it takes one picture, and this is fast. It takes, a few seconds. And your image is expressed in numbers, pixels, if you want to, in the digital imaging processing world. So you can see here on the bottom left that your cell is round. So the software is going to look at the roundness of your cell and the brightness, and it will compare the pixels inside your cells, which is the foreground, with the background. So that's perfect if you have cells above 8 micrometers. Then you will have an accurate counting. But what happens if your cells are below 8 micrometers? The problem is that the systems and the counters will have less pixels to compare, to look at the brightness of your cells versus the background. So if your cells are small, the variability is not always accurate. The total cell counting is accurate, but not always the variability. With the Luna 2, we improved the algorithm. We have an option which is called live cell sensitivity to improve that, but still it's not perfect. And if someone tells you it's perfect, well, run away, because that's not possible. And the other limit with image-based bright field cell counters is related to the dyes you're going to use. Remember, with the trypan blue or even with erythrosin B, the debris and the red blood cells are going to be counted as dead cells. So that's a limit. But if you're working with cell lines or clean samples, or if your cells are above 8 micrometers, that's perfect. You're going to have ideal counting with bright field image-based cell counters like the Luna 2. And here is the limits of I discussed just before. So if you are below 8 microns, you have inaccurate variability, and the solution is to go to dual fluorescence. So dual fluorescence cell counters include 2 technologies. So you have bright field, as with the previous instrument with the Luna 2, plus 2 channels of fluorescence. So you have 3 sources of light. And here we have 2 examples of dual fluorescence instruments that we provide, the Luna F and the FX7. For fluorescence cell counting, you're going to use fluorescence dye, so acridine orange propylium iodide. And as you can see here, it's going to count the expected cells. So here the monocytes and the lymphocytes are going to be counted when the red blood cells, and even the platelets, you can see the plates here, the platelets here in the background are not going to be counted. So the size, the cell size is not a limit anymore. And it's still, it is still a fast method because it takes 3 images, so in less than 30 seconds you have your results. But it also works with, let's say, more original samples. So if you take the example of chloroplast, those are big cells. You know, the question of the size is not an issue. But because chlorophyll is going to be bundled on the edges of the cells, it makes the count in bright field complicated. So for some samples, you know, fluorescence can solve the issue, even if the size is not a problem. When it comes to bacteria, we face even more challenges. The genome size and the cell size are around below 1 micrometer, and the shape. I mean, I don't know if you've seen bacteria under a microscope, but that's really Cocci, Bacillus, spiral can look like a star or a rectangle. But you also have the challenge of the membrane. The membrane, if you have the peptidoglycan cell wall for gram-positive bacteria, so sometimes it's a problem for the dye penetration. Other issues are related to the fact that bacteria are moving. So there is a Brownian motion inside the containers, and they don't settle down on the same focal plane as mammalian cells, and they are highly clustered. So you can imagine that the technology to count those bacteria are not only related to the instruments, but it requires a bundle solution, an advanced solution. And with the quantum, which is dedicated to bacteria, we got to optimize the loading buffer, we got to create a specific centrifuge, optimize dye, the instrument itself, and advanced algorithms to count the bacteria. So bacteria are very specific cells which require extra precautions. So if you're interested in that, we can make a presentation on that, but that would be too long to cover everything. So now we've seen that the combination of specific dyes and image-based technology is efficient to count cells. But the question is it enough? Is it sufficient to ensure that your counts are reliable? Well, you need to consider some parameters. And here I selected specific parameters that I consider as critical when choosing a cell counter. And the first point, we're going to go through a definition. Accuracy and precision. Accuracy is the proximity of results to the true value. So you have a target and you want to be close to that target. The precision, which is also called reproducibility, the precision is the degree to which replicates show the same results. Okay, so here this is accurate and precise. When it's accurate but not precise, you have a high CV, and this is how the results look like. You can be precise but not accurate, which is more rare, but can happen. Or what happens very often is that it's not accurate and not precise. So what you want is an accurate and precise technology. And at Logos Biosystem Aligned Genetics, it's been one of the key parameters which have been used to develop the instruments. So all our counters have been developed to be accurate and precise within the linear range of the cell counters. Here you have the Luna 2, and you see that the range, I mean, the linearity is perfect. So for the cell counters, you always have a linear range. Here, this is between 15,000 cells per ml and 10 million cells per ml, which is a nice range. But we could go below theoretically, but we decided to have this realistic lower limit, 50,000 cells per ml. It means 5 to 6 cells counted. You know, some companies say they can go below that, but it's when they count one cell, so it's not very statistically relevant. But anyway, but we can go even further. With the latest instrument we've developed, we can go down to a CV of 1%. This is unbeatable. Not other solution in the market, not other cell counter will go to such a highly accurate and precise instrument. And whatever the consumables we're using, I'm going to show you that later. But look, we're always below 5% when the average of instruments are, if they have 10% CV, that's great. I mean, for most of the instruments, our Logos products are below 10%, but you know, 1% is a big step. So one thing, remember, accuracy and precision, this is critical when you select an instrument. Another aspect is the field of view. So we take an image with image-based cell counters. The bigger the image will be, the more cells you will count, the more precise and statistically relevant you will be. So this is important that you make sure when you select a solution that you count a minimum of 5 square millimeter, okay, which represents about 0.5 microliter. Below that, and trust me, there are plenty of solutions in the market which are minimum 30% below that. Well, in that case, you better keep your hemostometer. Another question is, should we go with an autofocus or not? Autofocus is important when you do routine count, when you do lots of counts every day or every 2 days, or when you are in a production environment, because you don't want to spend time making the focus on the instrument. For R&D, it's up to you. It's less critical because you want maybe to look at different aspects of your cells. We propose both. For example, for the Luna 2, which is the bright field cell counter, it has an innovative technology for autofocus. So a liquid lens, which has no mechanical part. So the principle is simple. You have 2 liquids, water and oil, which can't miss. And they are separated by a small membrane. And when you apply the light, so some current, so it's going to move and adjust the focus. So no mechanical part, no noise, and it means this instrument makes no noise in the lab, which is quite rare. You're working in noisy environments. And this is an innovative and patented solution, and you still find it in a very standard instrument. Luna FL, you have the manual focus, that's for dual fluorescence, and Luna FX7, you have a high-end motorized autofocus, so you have the choice. The clustering is another aspect that you need to take into consideration. Here I took the example of the first and the last instrument we've put into the market. So you see that straight from the first instrument, so the Luna, the red Luna, we got outstanding declustering algorithm. So you can see that from the beginning, it was really important for online genetics to bring you the best algorithms to detect the clusters. And the FX7, the Luna FX7, the last instrument, goes even further, because most of the time, even if you're able to decluster live cells, it's highly complicated to decluster dead cells, and this instrument also allows it. Of course, the software is important. The software has to be easy to use, but also, so it means that if you have to spend 1 hour or 2 hours reading the manual before to use it, well, it's going to be complicated to get the best of the instruments. So it has to be intuitive. But you need also advanced functions, like the live cell sensitivity when you have small cells in bright field. The options like GFP transfection, we can provide that, it's provided with the Luna FL. Or if you're interested in, if you measure replicates, maybe you're interested in having the data combined directly, and that's available, and also the export solutions. With our cell counters, you can export everything. You can export the images, the raw images, the tag images, the PDF report, which contains all the data, the histograms, so you can see the repartition of your cells by size, the cluster map, so how your cells behave if they are alone or in clusters, and of course, consider free updates and virus-free environments. Another demand which was consistent and repeated by a number of customers was about throughput. Most of the counters allow to count one sample at a time. And most of our counters are counting one sample at a time. But we have also now new slides which are compatible with the Luna FX7, which allow you to go up to 8 samples at a time. But you see, even for the 8-channel slides, we still count a reasonable volume per chamber, okay? Remember the field view to have relevant results. So even with the high throughput channel slides, we can do it. Last but not least, quality control. For all labs, whatever you're doing, even if you're in academic, control your instruments regularly and ask if, how to test it with standard bits or validation slides. And if you're interested, there is also the IQOQ available. If you're working in, if you produce cells or if you're working in GMP environments, you will be interested by the compliance, the 21 CFR Part 11 compliance. The Luna FX7 has this option to make your counters compliant, but that will be another topic. And actually, we have another webinar next week where we're going to talk about the Luna FX7 and the compliance. That said, here is a question which is important. The question about pricing. You can imagine that the pricing depends on what technology, and I strongly invite you to ask for quotes to your local distributors. But let me answer part of this question. So you see we have a range, we have 7 cell counters. The prices will go roughly from $3,000 to $30,000. Okay, so of course you can have differences from one country to another, and maybe you have agreements with your distributors, local distributors, to have better prices. But you see that even the Luna 2, which has quite advanced technology on board, is one of the cheapest, if not the cheapest. And another thing is, see the Luna, the red Luna here, was the first instrument put in the market by Logos Biosystem. The Luna 2 was the second generation. Guess what? The Luna 2 is less expensive than the Luna. So new generation doesn't mean more expensive. Another aspect you need to consider are the running costs. The running cost is how much it will cost you every time you're going to click on count. Okay, so depending on the consumables you're going to use, as an average with us is going to be around 50-60 cents per count, which is already quite reasonable in terms of cost. But look, if you go with the high throughput channel slide, you can decrease by half this cost. And even better, you can even pay 0 per count, because we have also reusable slides, and they're compatible with all the instruments except the one for bacteria because of the difficulties I explained. That said, don't forget the quality of service and support. Ask for demonstration. Test with your own cells. Don't trust what we say during presentation on flyers. Test with your own cells. And we have highly educated and available distributors to support you in every single country. Ask for installation or training, even online training. I mean, those days with the sanitary situation can be a bit complicated, but we can provide online trainings. And also the after sales. We've seen so many companies that say, 1 week after the end of the warranty, oh, sorry, we can't repair. You have to buy another instrument. We don't, we repair. If necessary, we repair. If after 5 years, your LCD screen is broken because someone dropped on it, we can repair it, and at reasonable cost. But the last word is always the customer voice and the customer's feedback. And I have to say that we're very proud those days because we received a rare label of quality for the Luna cell counters. On Select Science, I don't know if you know this website, you can go and review products. And we have reached the highest seal of quality among tens of thousands of products. So that's only the 6th product worldwide, including reagents and service, to reach that level of certification. So take the time to review also what people said on our cell counters. And this is based on ease of use, the after-sale service, and the value for the money. So to finalize this presentation, just a summary and last tips. So if you work with cell lines, consider the Luna 2. It's fast, efficient, and affordable. If you're more orientated to R&D, consider either the Luna FL or the FX7. You have here 2 perfect technologies for R&D, and it's going to be perfect for complex samples like PBMC. And if you work with specific cells like yeast or bacteria, you have the Luna 2YF for yeast and the Quantum for bacteria counting. So that's the end of the presentation. I want to thank you for being there, and I'm going to look now at your questions.
Okay.
Okay, welcome everybody. My name is...
Hello. Okay, thank you very much for the presentation. Well, before we will go to the Q&A session, I will tell you about a little bit of a summary for this webinar in Bahasa. Jadi sebagai kesimpulan untuk presentasi pertama mengenai cell counter ini adalah, jadi cell counter dari Logos Biosystem ini dapat mendukung penelitian Anda dengan berbagai jenis cell counters. Jadi sampai 7 cell counter dari Logos Biosystem ini. Tentunya sesuai kebutuhan penelitian Anda masing-masing. Jadi jika Anda menggunakan sel bakteri, yeast, mammalian cells, ataupun cell lines, bisa menggunakan cell counter dari Logos Biosystem ini. Dan juga cell counter dari Logos Biosystem ini juga dapat mempercepat pekerjaan Anda dengan meminimalisir variabilitas antar operator dan juga subjektivitas setiap operator kan mungkin berbeda-beda. Nah, oleh karena cell counter ini adalah otomatis kerjanya akan meminimalisir standar deviasi yang disebabkan karena antar operator yang berbeda seperti itu. Dan mungkin langsung saja saya ingin mengingatkan juga apabila ada pertanyaan bisa langsung diketik di kolom komentar baik itu dari Zoom meeting ataupun dari YouTube yang nanti akan saya sampaikan seperti itu. Thank you very much for the presentation. Now we will continue for the question and answer session. So we have the several questions here. Probably first of all from Mr. Budi Wicaksono from UB or Brawijaya University. The first question is, in counting cells using hemocytometer or hemocytocounter, is there a minimum standard of cells to be counted? Yes, that's the first questions from Mr. Budi Wijaksono. Kelly, are you still there?
Yes, and I will answer the questions. Yes, that we have the cell counting range that is optimal for our cell counter. The minimum cell concentration you need to prepare is 500,000 cells per ml. And then the maximum is approximately 5 to 7 million cells per ml.
Okay, thank you very much for the answer. For Mr. Budi Wicaksono, is the answer enough or you have further questions? Or If you have further question, please unmute the recording, the sound, so we can hear your voice.
And
But
then
if there's no further question,
I am seeing
okay.
the chatting box that the customer Yenny used, and then this person asked that what caused the clustering of the cells. The region of the clustering of cells are various. Sometimes if cells are old enough, they are clustering together. Or when you harvest the cells, you need to detach the cells using the trypsins. If you did not treat enough trypsin, the cells will be aggregated. And the other thing is that you need to do enough vortexing and pipetting. So if you did not perform enough vortexing or pipetting, the cells will be clustered together.
Okay, so the pipetting is important, right?
Yes.
Oh, okay. Thank you very much. And yes, that's. Let me check the YouTube first for the other questions. Well, yes, I think that's all for the questions. We have another question, I think. From Susiana Melanie, what is the difference between hemocytocounter and flow cytometer?
That I think the customer asked about the manual cell counter hemocytometer or automatic cell counter or flow cytometry. Okay, that I will explain about all the three methods.
Yes.
The first of all is that this manual cell counting, you are using hemocytometer using microscopy. And I think we already know all about this, just seeing the microscopy and then count each cells. Using our automatic Luna cell counters, we are using the almost a similar optic system of your microscopy. We find the cells, each single cells, and by image based, so we count the cells. But if you choose the flow cytometry, it uses the signal of the cells or tagged protein or antibody fluorescence signal, so that it uses the impedance method to count the cells. But the main difference between hemocytometer or our cell counter and flow cytometer is that flow cytometer is extremely expensive and also really difficult to use the machine. Also, consumables are very expensive compared to our Luna cell counter consumables.
Okay, thank you very much for the clear explanation, Kelly. So is there any other questions for the session? Cell counter that will help your research and QC. Okay. If there's no further questions, we can presentation regarding the Selena imaging system that will be held.
Okay, then I will start the presentation of Selena imaging system. If you have any questions about the cell counter or Selena imaging system, please leave the message in chatting box, or you can also send the email to the PT Plasma. Okay, then is that okay to start the Selena imaging system webinar?
Yes, Kelly. But Kelly, sorry to interrupt you. But can you please turn on your video first before you start your presentation?
You mean the video?
Yeah.
Okay.
Yes.
So we all can see you.
Yes.
Okay.
That's
all right.
Is it okay?
All right, Kelly.
Sure.
Thank you.
Okay.
Okay, you're welcome. And then I have a colleague next to me, and so we can see together. He is helping me today. Okay, let's begin. Okay. Okay, again, hello everyone. This is Kelly, and then I'm the product specialist at Logos Biosystems, a part of Aligned Genetics. First of all, thank you very much for your valuable time to attend this webinar. And I'm very happy to electronically meet you all. And I've seen that the COVID situation in Indonesia is really good. So that I really do hope that everyone here and colleagues and family members are all safe. And okay, the Korea, the situation is not really bad at this moment, but confirmed number is slowly and gradually bit increasing. So we are extremely careful about the virus infection. So virus cannot stop us, so we must keep going. So today wait a minute. Today I will talk about the imaging system, introducing our imaging system, the Selena S first, and then Selena X, the features of each product and the application of each two systems. You may heard of the word of high quality image almost everywhere. It is very important for everyone who is using microscopy, because image is upstream process and analysis is downstream process. The analysis does not work well if the image quality is bad. So the question is that what is high quality image and then how to get this? When we say the high quality image, it means that the signal level of image is high and background noise level is low. This is so-called SNR, the ratio of signal power to the noise power. The ratio higher than 1 to 1 indicates that the signal level is higher than noise level. As shown in this picture, if the score of SNR value is higher, means that quality of image is good. The image is very clear and vivid when the score is 15, but the image is blurred and unclear when the score is 1.5. The next question is how can you get the high quality image in biology? I will introduce 8 tips to get high quality image today. The sample preparation is critical to ensure the high quality image. The adequate density of the cells should be prepared without any contamination, because the foreign particles or debris can negatively impact on the image quality, and they can also interfere the focusing of the cells. Choosing of the fluorescence-proof staining protocol are also very important. It is important to increase the fluorescence signal level against the background. So you need to find the optimized staining protocol of your cells. Usually, you can easily find those information in internet. You also need to carefully choose the sample vessel. The bottom of the thickness of a sample vessel needed to be considered depending on what type of objective lens you are going to use. So the thickness of a glass slide is about 1 milliliter, and the cover glass is 0.117 milliliter. And most of tea flasks and dishes and multi-plus plate, especially made with the plastic, have 1 milliliter bottom thickness. If you choose the wrong vessel, the microscopy cannot find your cells. If you are going to do cell-based assay, the proper segmentation of the cells is needed. It is really difficult to analyze the image if the cells are aggregated, or there are too many cells, or there are too small number of the cells. You need to check whether your microscopy can focus the adherent cells or floating cells. If the cells are floating around, you will need to prepare the microscopy which can do the Z-stacking. Also, you have to consider what type of the cells you want to observe, such as common cell lines or primary cells or stem cells. If the cells are growing over the 2 layers, that you cannot focus them using the simple microscopy. Choosing the proper equipment is also very, very, very important. In terms of fluorescence microscopy, excitation and emission spectra of fluorescence proof or protein should be matched with LED light sources. And also you can choose the auto focusing function if you are looking to use automation system. It is very useful when doing the live cell imaging especially. Autofocus is also very useful for fluorescence imaging because it can be accomplished by using the signal from the included fluorescent label. Appropriate image acquisition settings are also critical to obtain the meaningful and quantifiable data and images, which is suitable to use for the publication. These settings typically include excitation light intensity and camera gain and integration time. For example, the light exposure setting affects to the pixel saturation and background level, so it will increase the total acquisition time. Long-term kinetic live cell imaging typically requires some control of the environment conditions to maintain the sample viability. The temperature and atmosphere conditions are the main consideration. For example, you need to incubate the cells which can maintain the 37 Celsius degree and 5% CO2 gas concentration for mammalian cell culture. And you need high quality incubation system to allow live cell imaging without any sacrificing sample integrity over the long period of kinetic reach. Okay, then I think that some messages are coming up. Okay, all right, I will continue. When you prepare the samples and machines perfectly, there will be some difficulties. This is the component of the conventional fluorescence microscopy. There are several different components which should be handled very carefully. Also, it takes time to turn on and turn off the entire system. Sometimes it takes more than 30 minutes and sometimes it takes more than an hour. The problem is that you have to put these components in the darkroom with you. You have to see the strong fluorescence light in the darkroom and for several hours, so your eyes will hurt. So worst case is that your colleagues will forget you. So maybe they lock the door and finally you will be left in the lab alone. It sounds a bit complicated because you have to consider so many things to just see your cells. But if you're lucky, you now have our system, Logos Bio. We provide two different types of the fluorescence imaging system. One is Selena S. This is a manual type of a microscopy, which is economical model for scientists who have the limited budget. If you are looking for the automation system, there's higher grade, which called Selena X. This is also economical model compared to other big players. I will shortly introduce what functions we provide and how it can be used for your application. Let's see the Selena S fluorescence imaging system first. Selena X looked quite simple, which means that it is extremely easy to use. You can even guess what you have to do without reading the entire user manual. The light intensity can be adjusted from the upper part of the machine. And the stage is located in the middle. And there are two knobs for focusing. One is coarse and the other is fine focus. And you can easily change the objective lens by moving the turret. You can accommodate total up to 5 lenses. Also, you can use minimum 1.25x magnification up to 100 magnification. There is onboard storage space, which is 128 gigabyte SSD storage. There are also USB ports, so you can easily extract the data from the machine. You can install total 3 LED filter cubes and 1 bright field light source. Moving the stage, the focusing and changing the objective lens and LED filter cubes are extremely easy and almost the same as common microscopy. We offer different type of the vessel holder. You can use a tea flask and dishes, multiple plates, and glass slide as well. By using the universal holder, you can use any type of the vessel you want. The specified holders are useful for live cell imaging because the sample vessels should be fixed to capture the exact same location. The Selena S is the economical fluorescence microscopy, but it proves several functions you will need: bright field and phase contrast, fluorescence, image analysis, G-stacking, cell counting, and time-lapse. First of all, you can perform traditional bright field imaging. By just changing the annealing at the top of the machine, you can also do the phase contrast imaging. This picture shows the same cell with traditional bright field and phase contrast. You just need to prepare the phase length. As you can see in the picture, the phase contrast imaging shows so many cellular structures, more visible than bright field microscope. It is good for quantifying the cellular structure and component, which does not use fluorescence. You can also do the beautiful twinkling fluorescence imaging. Okay. Okay, sorry. I will repeat again. You can also do some fluorescence imaging by using the Selena S. These are the examples which we actually use the Selena S and obtain the image. After took the images using different color LED filter cubes, you can overlay them and see with the pseudo color. We use the BPAE cells labeled with the palladium for red, alpha tubulin for green, and DEPI for blue. And we use the 40x magnification lens. The image is very clear and beautiful. To analyze the data, if you are using the traditional microscopy, you actually have to learn some complicated software, which we biologists do not like. You don't need to pay extra money for it if you have a Selena S. These functions are already included in the machine. You can measure the length and area of interest, and also you can see the histogram of light intensity of each fluorescence signals. You can also add annotations such as a scale bar or text or other figures. After taking the images, you can also edit the pictures similar as your cell phone. You can adjust the brightness and contrast of the image, so you can make the images more beautiful. If the cells are floating around and sample is thick, you need to capture the images from the different G positions. These gray images are taken by using G-stacking function. The sample is lava of the zebrafish. These images were taken by the customer from Malaysia during the demo. It was the first try to them, but they got the very good image very easily. The next image shows the G-stack of the brain tissue image. You can see the neurons and cortex from the different G-axis. At the first session of webinar, we introduced the cell counter. You can even count the cells using Selena S. The fluorescence cell counting is not available, but the bright field cell counting can be done perfectly, almost the same as our Luna cell counters. The machine looks a bit simple, but yet it provides sophisticated functions. This is a simplified diagram. Okay, this is also the live cell imaging is available, accommodating the on-stage incubator. This video shows that how you can prepare the cells and incubations for the live cell imaging. We use the HEC 293 cells and then do the imaging for the wound healing research. I will show you this video with some big screen later. So, okay, this is a simple diagram of the cell culture and imaging. You need to monitor the cells as they grow, and then you have to count the cells. The routine work is done for various purposes, which means that you can do lots of images by doing the cell culture. You can observe the cell structure. confluency and also growth of the stem cells, measure the transfection efficiency, and monitor the transgene expression, protein localization and co-localization. Also, you can do wound healing research, hypoxia, and many other applications. These applications can be done by different imaging mode of Selena S. For example, bright field imaging is good tool for observing the cell growth, cell structure, and healthiness, and monitoring of the cell confluency. It is also good to observe the zebrafish, as mentioned previously, excellent for cell counting as well. Please note that the fluorescent cell count is not available. To do this, you will need our LUNA-FL fluorescent cell counter. By using the fluorescence imaging mode, you can measure the transfection efficiency, transgene expression, and GFP expression as well. You can also use the different color to see the tagged protein of the interest. Also, you can do the protein localization and co-localization. Also, you can identify the structure of the cells or tissues. You can also observe the pluripotency of the stem cells. Let's see some other example images, what our Selena is capable of. The stem cell colony differentiated into the neurons by using different colors. The stem cell protrusion can be also observed using Selena S. So you can study the adipogen differentiation. By using the fluorescent tags, you can check the protein expression level as well. You can also observe the multinuclei in bone cells. Microscopy is also widely used in food research. In this picture, you can see that the meat is contaminated by Salmonella. In the second picture, you can see the protrusion in adipose tissues. The neuronal protrusion can be also clearly detected by Selena S. Recently, the organoid and spheroid research is very popular. You can do a lot of structures. Okay, wait a minute. This is a video. Okay, now you can see that. Mostly they are used for drug discovery. Unlike the cells, organoid and spheroid is thick, usually from 100 micron to 1 milliliter. So it is really difficult to capture the images of whole area in fixed G position using the simple microscopy. In this case, the G-stack is very useful. This is the video clip of the G-stack of the spheroid tagged with GFP and RFP. The second video shows that the spheroid with 1 milliliter thickness. They are labeled with RFP like this. You can get the video clip and merge the image and each image as well. Okay, the video is not working perfectly. Okay, wait a minute. Okay, it's not working. Okay, anyway, we saw that the G-stacking worked wonderfully for the left image. So let's see the other application. By accommodating the Ostage incubator, you can also do the live cell imaging. For example, you can observe the mitosis of the cells, proliferation, angiogenesis, or chemotaxis, or hypoxia, even for spheroid physioxia, migration, and wound healing studies a lot. This is the video clip of the wound healing. We used HAC-293 cells. Let's watch this. We used the 10x magnification lens. The images were taken for total 10 hours with a 10-minute interval. And the cells were cultured in 35 milli dish. You can see the very nice video of a proliferation of the cells. You can also check the other videos from our website. If you want to high throughput automation system, we can also help you. The automation system is not also complicated if you work with us. These are all the components you need to do automated high content imaging. You need a monitor to see the machine. and main body, which is actual microscopy. The door is easily closed and open, and there is a condenser inside the machine, and there is a motorized XYZ stage, which is automatically worked. Also, there is a power button, just press it. And then we need a computer, which is just the same as your desktop computer. And also we need a controller to actually give the power to the machine. These are the several options you can choose. Depending on your application, you can choose either image-based autofocus or laser autofocus. You can decide whether you are going to use a phase contrast condenser or just a simple light condenser. If you want to see the tissue section for histology, you need to use the color camera to see the H&E staining and other stainings. There are 12 commercialized LED filter cubes, and then you can also use special custom filter cube if you want. Total 4 fluorescence filter cubes and one bright field filter cube can be installed in the machine together. As the same as the Selena S, you can use the objective lens from 1.25x magnification to 100x magnification. There are also 12 different type of the vessel holder. So you can use the different type of the vessel, such as glass slide, multi-wave plate, dishes, and tea flasks. This is very important feature for you, that we provide the complete version of the imaging acquisition and analysis software without any additional cost. This software will be already pre-installed in the computer, so you do not need to pay single penny or single dollar for the software. You can perform fully automated imaging acquisition and analysis by using Selena X. It is simple. Just put the vessel holder and vessel on the stage and then select the area and just a few clicks. That's it. The diagram shows a little more details. First of all, acquire the images automatically. Okay, seems that somebody turned on the microphone. Could you please mute your microphone?
Oh, yes. Yes. Wait a minute, we're working on it.
Okay. Okay, I think it worked. Okay, you can, this diagram shows the little more details. First of all, acquire the images automatically, and then analyze the image itself and get the data. Then analyze the obtained data and then plot your result. This shows the real screen view of vessel. The left one shows the 96 well plate. You can even select one small field from the one small well. You can use the minimum 6 well up to 384 well plate. And live cell imaging, G-stacking, stitching, merging, and are all available. The right picture shows the real screen view of the glass slide. You can put the slide face up or face down. The stitching function is really useful when you are doing the imaging of the slide sections. You may also use it for tissue microarray. I will show you the example later. The slide shows all the functions what the Selena X has for imaging acquisition and analysis. There are too many functions, so I will simplify and show some important application today. I will show the real screen of the Selena X image acquisition. This is the video. Okay, wait a minute. Let's see, it is working, not working. Okay, this is not, oh, sorry, the video is not working here. But then you can check from our website, or that we can send you the file. Then I would like to show that this is a real screen of the hematoxylin and eosin stained tissue section. As you can see here, the image is quite clear, but then we make it a little bit darker to to facilitate the imaging speed. The next one is the 96 well imaging. It should work. I'm sorry. Let me try it. Okay. I can also send you some files to you, or that you can also check from our website. I want to say that, okay, we prepared, if you have the cells from all the 96 well plates, then and stained with the 3 colors like DEPI blue and the DEPI to see the nuclei, or you can stain the AO to see the cytoplasm, and then you can also stain the mito tracker to see the mitochondria, and then these will illuminate in the 3 colors, so we can capture those images using DEPI, GFP, and RFP channel, and get the image from the whole entire 96 well plate takes less than 4 minutes. Yes, 4 minutes. This is really fast. And I can give you a little tip on how to facilitate the imaging acquisition speed. It takes too long to acquire the image if you find the focus from all 3 channels. In that case, please find the focus in DEPI channel first, and then do not set the focus for GFP and RFP, because it will be already good because we find the focus in DEPI channel. And then okay, and then the other tip is that you need to decrease the time of the light exposure to facilitate the imaging speed. So in last slide, so that is why that we made it a little bit darker. If you are interested in live cell imaging, yeah, this is really popular in the world at this moment. Everybody is doing that. You can add on the incubator for both Selena S or Selena X. Our incubator has a heater on the glass to prevent the condensation. As there is a glass protector, it is not easily broken. And this glass protector also acts as double windows to prevent any environmental disturbance. And you can use the multi-plate dishes and chambered slide or cover glass by using the adapter. You can also monitor the cell culture environment condition in your PC. This is the real image, and the bottom is the video. Let me check whether it is working or not. Okay, it is working. Using the on-stage incubator, the live cell imaging can be done. You can get each individual images, and you can create the video with just a few clicks. This is a video of the HEC 293 cells cultured in 96 well and took the images for total 42 hours with 3-hour intervals. After acquired images, the software finds the boundary of the cells and measure the area, then analyze the data. You can also get the cell growth graph as seen in this slide here. And this is the example of the application of wound healing. Okay, this migration of the 2D sheet expansion occurs in diverse biological processes, including the embryonic morphogenesis and cancer metastasis. The most advantageous of this assay is that the cell migration rate is simply measuring the increased cell area in the cell-free gaps of the specific chemical attractant or gradient chambers. The assessment for guiding to investigate the cell migration by using time-lapse acquiring images. Using the analysis pipeline, wound healing process was successfully measured, and the analysis of the measurement of wound research resulted in plot representing the wound closure to the migration over 16 hours. The tube formation assay is widely used for in vitro assay to study the reorganization stage of the angiogenesis. In vitro assay of angiogenesis typically study the behavior of endothelial cells within a controlled environment. It is really beneficial to study vascular and developmental biology, especially for angiogenesis investigation. it should be studied in very strictly controlled environment, which means that the incubator should control the CO2 concentration and temperature very accurately. This video is an example of the tube formation assay, and then culture the Hubeck cells on 96 well plate with the Metriger basement in the incubator. Image acquired for total 6 hours in bright field imaging mode. After acquisition of the images, analyze the area and perimeter of the tubes and shows in graphs. And this is another bright field images, that image analysis. We have seen the cell growth and migration for wound healing assay, but there are also many applications available when we monitor the cell death. Cytotoxicity assay are a crucial step in screening and developing the therapeutic anti-cancer drug or any other cancer discovery. To study in vitro cytotoxicity, we treated HeLa cells with the different concentration of the camptothecin, which is the type of the drug. The cells were imaged automatically at 2-hour interval for total 16 hours with the Selena X. In this study, we analyzed the effect of different concentration of camptothecin on compliance using the Selena X. There's some limitation of the bright field imaging when doing the cytotoxicity assay. By using the fluorescence imaging, more wide applications are available, especially for broad spectrum of the drug in the cell-based analysis. It can measure the cytotoxicity in vitro, evaluating the cell membrane integrity after the exposure to the drug. Also enabled to calculate the maximal inhibitory concentration of the non-linear regression of the agonist, as well as variable in vitro values in the batch process. Additionally, the endpoint measurement of the CPT toxicity is available by applying Hodges dye. Among many molecular biology procedure, the gene expression is the most common molecular technique to transfer an exogenous gene to host cells. We performed the transcription efficiency assay using the Selena X and demonstrated that the high content analysis of nuclei localized gene transfecting to HeLa cells. The cells were prepared in 96 well plate, and then cells were applied the GFP plasmid DNA transfection procedure. The transfection efficiency can be plotted and clearly demonstrate the increment of the efficacy on the amount of the plasmid expected. More sophisticated imaging and analysis are available using Selena X. This is a process of the human iPS cell differentiation, can generate the several different type of the organ. The upper image shows that shows the eye. The bottom image shows the intestine. In this study, it shows that the advance of 3D cell culture system to improve drug target validation, and it leads to optimization, which make a significant impact in disease modeling and drug discovery. To do this, just a simple light microscopy is not helping, because the average size of the organoid for research is about 500 micron to 1 milliliter. You cannot see the entire layers of organoid using simple microscopy. For organoid imaging, you really need maybe an expensive microscope. The confocal microscopy might be very good, but it is really expensive. So by investing a lot less money, then you can get the very good organoid imaging. Here is the example. We made the colorectal organoid with 350 micron thickness. It is performed G-stacking, and you can see very clear image of each layers. You can set the interval of the layer narrower or wider. If you stain the organoid with the several different staining dye, you can overlay the images together. And there's common challenge we are facing. that when we grow the cells, and then we can see the effect of the drug treatment. Organoids are not thin and they are growing three-dimensionally, which means that you have to move the focus to find the organoid by moving around the z-axis. In this case, you will need a laser autofocusing, which enables the find very clear focus during organoid culture. We have treated the different concentration of the drug, and then we saw the effect on the organoid, and they calculated the dead cell population by reading the intensity of red and green signal, and then plotted the dose effect on dead cell population. Not just about the fluorescence and three-dimensional organoid images. You can also do very basic microscope work by using Selena X. This is a very simple example of the H&E stained sections, which are typically used for evaluation of the pathological process. The sample is paraffin section of the brain tumor biopsy. The tumors can be seen very clearly from 4x to 40x high magnification. This is the example of the stitched images. Some people want to get the image of the large specimen with the high magnification, but if you use high magnification, there is a limitation of area you can see. Also, a lot of data will be created if you use high magnification. And sometimes we want to see the overview of a large specimen. In this slide, you can see that the perfectly stitched one big image that we stitches many individual images. You can navigate them, zoom in and zoom out of stitched image. If you want to see the certain area very clearly, you can just simply zoom in by moving the mouse wheel. The combination of the bright field and color camera, it will create high quality image good enough to publish. Also, one big stitched image can be analyzed by using our software. I showed the bright field stitched image that you can also use the fluorescence images for stitching. Here is the image of a mouse brain with the fluorescence labeling. The images were taken using 20x magnification lens. We used the 2-year-old mouse brain. When you magnify this, you can see that there's no missing area or tilting. I cannot find whether this is just one big image or stitched the several images. You can see more details of different color of fluorescence label. Also, you can see the depth signal and nuclei of the choroid plexus area. People conventionally make the paraffin sections for the pathologic and histological evaluation. The high throughput tissue array can be applicable to Selena X system. Many laboratories want to make the simple screening of entire immune organs, brain, nerve, and multiple organ as well. Sometimes get the tissues from different organs or get several tissue sections from different area of one organ, or can get the same area of tissue and compare the positive and negative effect of treatment in that area. The individual tissue sections can be plugged in and reassembled in one slide and screening using Selena X. You can modify the setting of the vessel, so you can acquire the entire very nice images of each tissue sections on one slide. This is a multi-view of the tissue microarray slide in Selena X. Each individual sections can be magnified using high mag objective lenses, and then they obtain the data of the targeted protein. In this case, it is immunochemical stained the target and scored their level of brown spot, not just about the image acquisition, but also analysis of brown spot is available by scoring pixel number of brown spot. Scoring of high number of brown spot versus of a lower score in neighbor area. You can easily handle and represent the data. In summary, Selena X is very handy and user-friendly microscopy. and I introduced some examples of application of Selena X: cell growth, cytotoxicity, wound healing, and tube forming assay in bright field, and cell counting, cytotoxicity, cell transfection, and organoid in fluorescence imaging. There are also a lot of other applications you can do using Selena X, such as beta-galactosidase assay, calcium efflux assay, or phagocytosis, and others. There are already many sophisticated scientists using our Selena X system. I just picked up the 4 reviews from Select Science website. Many people found that Selena S is extremely very easy to use, and even for beginners for the fluorescence imaging system. The image quality is very high, so that it is good to use for publication. The image quality is high, and also the Selena X is a perfect combination of the high content and high quality imaging. And you don't really worry for becoming the first customer, because some scientists are worried to be the just first users of the new machine, but the Selena S and Selena X are not the new machines. There are already more than 100 papers published using Selena system. There are too many, so I cannot write down here the entire list in the slide. Our Selena system has been used for various fields of research and improved the image quality by scientists worldwide. You see the very good journals here, even the Science and Nature. You can check more citations in Google Scholar or in our website. Thank you for your attention. And do you have any questions? Okay, I see that.
Okay, thank you very much, Kelly, for the great presentation. Well, actually, we have the several questions.
Okay, yeah,
for the Selena you already saw.
Okay, I will check. I will check the chatting box. The question.
Well,
the first question is from Bajar from UB.
Yes.
Yeah,
Yes.
Can this equipment good for cell growth or only as camera for cell culture? I think you already saw that. Yeah, it is good for cell growth as well. And the next question is sorry, my pronunciation. What this tool can count cells from organ tissue, either normal, necrotic, not blood, or tissue collagen. Okay, you can stain the nucleic acid using DePi or AO or PI, and then we capture the signals from the nucleic acid, and then we found the boundary of the cells. So in that way, we can count the cells, whether they are alive or dead, because we can also set the threshold of the light intensity.
Okay, Kelly, wait a minute, but can I also translate in Bahasa for your answer? So it will be convenient for the listeners, right?
Okay, sure.
Okay, untuk pertanyaan pertama dari Pak Fajar Sidik Permata, jadi itu memang juga sudah bisa digunakan oleh untuk cell growth atau pertumbuhan sel, seperti yang tadi sudah dijelaskan di presentasi, seperti itu. Dan untuk Ibu Endah Wulandari dari FKUIN Jakarta, ya itu dapat juga digunakan untuk pengecekan dengan menggunakan normal atau necrosis cell, dikarenakan ada beberapa fluorescence ataupun dye ataupun pewarna seperti AO, iodium propidium dan iodium propidium atau IP, AO, IP dan juga DAPI yang dapat menempel pada sel-sel dan juga bisa membedakan sel hidup dan juga sel mati. Selain itu juga terdapat threshold di dalam Logos Biosystem mesin ini, baik itu sel kantor maupun di Selena, jadi dapat terdeteksi fluoresensi minimum dari sampel yang Ibu deteksi, seperti itu. Okay, Kelly, I finished translate into Bahasa, so you can continue for the questions and answers.
Okay, thank you so much. And then the other question is also from Fasar Siddiq. And
Yes.
how many hours and days grows inside this equipment? Okay, you can want to grow the cells as long as you want, because there is some chambers that you can add on the water inside, and so keep the moisture inside the chamber. And Azalea, do you need to translate it?
Yes, yes. Okay, I will translate the answer. Well, jadi untuk inkubatornya itu sampai berapa lama bisa digunakan inkubasinya? Nah, itu tidak terbatas karena memang inkubasinya itu jadi dapat menahan sesuai kebutuhan user. Misalnya ingin diatur kelembabannya atau suhunya, dan juga ataupun ada temperatur ataupun gas-gas seperti CO2 ataupun oksigen seperti itu. CO2 atau oksigen.
Boleh menambahkan kan?
Oh iya,
ini
boleh.
Integrasi kan dengan CO2 ya? Ada integrasi dengan CO2 ya?
Ada apa, Pak? Maaf?
Ada kan kalau kita harus growth cell, we need carbon dioxide. So it's integrated with the equipment or spirited with the equipment of the carbon dioxide.
So Kelly, oh yeah, Kelly, did you hear the further questions regarding the incubations? So the questions is the carbon dioxide for the incubation? If we need the carbon dioxide for the incubation, we need is the instrument separated or integrated into the machine? That's the question. Is it correct, Mr. Fajar?
Yes. Thank you.
That's the questions.
Okay, you can think that it is almost the same as your CO2 incubator. You need to prepare the CO2 gas tank, and then you can simply connect to the incubator. It is the same system as your big CO2 incubator.
Well, excellent. Yeah, interesting with your question. Thank you very much.
Okay, thank you very much for the answer.
Okay, the next question is from Suri Mustaina. Okay, can this tool be used to trace the metabolic traces of some substances in various body tissues? Okay, it depends on then which metabolic traces you want to track. If you stain if the certain metabolic substances can be stained using the staining dyes, yeah, we can track the signal of the. I think the fluorescence would be better because we can check the light intensity that is from the metabolic substances.
Ya, saya akan menerjemahkannya. Jadi untuk Ibu Sri Mustaina, apabila metabolic traces yang akan dicek oleh ibu itu dapat berikatan dengan dyes ataupun zat pewarnanya, itu tentu saja dapat juga didetek oleh mesin ini, seperti itu Ibu. Jadi dapat juga kalau misalnya ada ibu membutuhkan atau ingin mengecek metabolic tracesnya ini atau beberapa substansi dari beberapa jaringan tubuh, tapi jaringan tubuh itu memang bisa berikatan dengan dyes seperti AO, ID, dan juga DAPI, itu bisa juga dicek fluorescensinya seperti itu Ibu. Yes. Thank you very much for the answer. Yes.
Okay, the next question is that I will just read in English that we do FUMIPA. Okay, it is it is about the cost and asking about the demo, and our PT Plasma will answer you the question later after this webinar. Okay, and the other question is also from the same person, that what is the light source of the light? Is it LED or mercury? Yeah, the light source is LED.
Jadi sumber cahayanya itu berupa LED ya, Ibu.
Okay, I think this is the last question from Dr. Desi Arisanti. Okay, I'm interested to see visualize the cell transfection by mRNA. Can this tool to trace position of mRNA in the cells which transfected into eukaryotic cell lines? Yes, you can use the fluorescence tag protein. And then if the transfection was successful, yeah, you can trace it. Also, it can calculate an analysis of the transfection efficiency.
Jadi untuk Ibu Desi Arisanti, ya bisa untuk dicek apabila hasil transfeksinya itu berhasil atau tidak, dan juga bisa dicek juga ya hasil transfeksinya intinya bisa dicek terhadap menggunakan alat dari Logos Biosystem ini, seperti itu Ibu. Ya, okay. I think that's.
Sorry, Kelly and Kazel. Maybe we have, because we still have more times. Maybe you skip the first question from Putu Erika.
Yes.
Yeah, the question is, if we use tumor cells, can we detect mutated versus wild-type gene?
I'm sorry, could you repeat again, please?
If we use tumor cells, if we use tumor cells, yeah, can we detect mutated or versus wild-type gene?
Kelly?
Okay.
Can you understand the question?
I heard that if the customer used two different type of cells, and then what was the next?
Well, actually, but can you hear my voice clearly?
Yes, yes.
Okay. So our customer or our visitors is asking about Luna FX7.
Okay.
So she's asking that if she used tumor cells, can she detect the mutated or the wild type gene in that sample, for example?
Okay, FX7 is just a simple cell counter, just counting cells, and it cannot distinguish whether the cells are mutated or not because it just counts cells.
Okay, I will translate first. Kepada Ibu Putu Erika dari UNUD, untuk Luna FX7 ini, dia tidak bisa membedakan apabila sel tersebut mutasi ataupun wild type karena proses ataupun tipenya ini adalah hanya untuk mengecek dan juga menghitung sel seperti itu. Tapi Luna FX7 ini bisa mengecek apabila sel itu hidup atau sel itu mati seperti itu. Yeah, that's for the questions for Putu Erika from UNUD. Yes. Okay. Thank you very much, Kelly, for the excellent and the great presentation.
Thank you.
And also, probably before closing, I will say a little bit summary for the Selena.
Okay.
In Bahasa, of course. Jadi Selena ini adalah suatu sistem pencitraan yang sudah terintegrasi dan juga dirancang untuk mengakuisisi dan juga menganalisis gambar dengan cepat tentunya. Nah, untuk Selena ini juga akan memudahkan pengambilan gambar dengan resolusi yang tinggi dan juga sangat berkualitas. Selena ini juga sudah dilengkapi dengan perangkat lunak yang canggih dan juga sederhana yang dapat mendukung berbagai macam pewarnaan seperti multicolor fluorescence imaging, bright field imaging, phase contrast imaging, live cell time lapse imaging, dan juga Z-stack imaging. Nah, untuk yang live cell imaging ini juga terdapat inkubator untuk mendukung proses inkubasi dari sel yang Bapak Ibu ingin inkubasi seperti itu. Jadi sambil inkubasi sambil dapat juga dimonitor sekaligus. Dan juga Selena ini pengaplikasiannya sudah banyak digunakan oleh seluruh peneliti di seluruh dunia. Dan juga pengaplikasiannya dapat digunakan sebagai cell growth, juga untuk cytotoxicity, wound healing, dan juga cell counter tentunya. Okay, thank you very much Kelly once again for the excellent presentation and the great answer.
Thank you so much for
Thank you
all
so
your
much.
time.
Yes.
Yeah.
And unfortunately, that's all the time you have for the question and answer session. And probably if you still have the further questions, you can email to PT Plasma Unitek Global directly or to Logos Biosystem directly. And then for the remaining questions that weren't answered during the live Q&A session will be followed up by email, of course. And once again, I'd like to thank to Kelly and all the attendants for this webinar. And after this, I will pass it over to Oscar as a moderator. Please, Oscar.
All right. Thank you, Kelly. Thank you very much for your
Thank you.
for all the information about Selena and the cell counter as well. Kelly, before that, can you please stop the share screen?
Okay.
I want to give some information to the audience here.
Okay, okay, wait a minute.
Okay.
Okay.
Thank you, Kelly. Sorry. Baik. Terima kasih Bapak, Ibu, dan seluruh rekan yang telah berpartisipasi pada webinar kita siang hari ini, hingga sore hari ini. Dan kita sudah berada di penghujung acara, namun sebelum itu saya ingin memberikan informasi menarik bagi seluruh peserta yang hadir pada webinar kita kali ini, baik di Zoom meeting maupun di live video kami. Kami akan memberikan special offer atau penawaran spesial berupa diskon sebesar 30%, dan ini hanya terbatas hingga tanggal 20 Desember 2020 untuk seluruh peserta yang hadir pada webinar pada siang hari ini. Nah, untuk item yang kami berikan penawaran ini berupa automated cell counter atau Luna 2 ini. Nah, jadi Bapak Ibu dapat menghubungi langsung kontak yang sudah kami berikan. Tadi juga sudah ada yang bertanya di chat, di grup chat ya. Ada yang bertanya harganya berapa nih, harganya berapa. Nah, boleh langsung menghubungi tim kami dari PT Plasma Unitek Global sebagai distributor resmi untuk Indonesia. Nah, oke, saya ingin berikan info terakhir juga bahwa mungkin ini akan berkelanjutan dari webinar kita siang hari ini, bahwa kita akan melanjutkan atau memberikan webinar kembali pada tanggal 25 November 2020, satu minggu ke depan. Kita akan mengadakan webinar lagi mengenai cell culture solutions and bertema Cell Culture Solutions and Partner for Your Research and Industry. Nah, ini kami juga akan mengangkat tema dari salah satu produk unggulan kami juga, yaitu Panbiotek yang menawarkan serum, media culture, dan sebagainya. Nah, jadi ini akan menarik sekali jika Bapak Ibu ingin mengikuti webinar selanjutnya ini, dapat langsung menghubungi tim kami juga untuk melakukan pendaftaran, atau Bapak Ibu dapat juga mengunjungi sosial media kami. Sosial media kami di antaranya ini ada Instagram kami, PT Plasma Unitek Global. Nah, untuk Bapak Ibu yang tadi ingin melihat video-video yang diberikan oleh pembicara tadi, dapat mengunjungi websitenya, yaitu logosbio.com. Nah, seperti itu. Baik, karena kita sudah berada di penghujung acara, sekali lagi saya mengucapkan terima kasih untuk seluruh hadirin. Sampai jumpa di webinar berikutnya. Selamat sore semuanya.
Selamat sore, terima kasih.
Okay, thank you very much for attending our webinar today. Thank you.
Oke. Nah, karena masih semuanya belum live, mungkin yang belum meninggalkan Zoom meeting ini, kita mau screenshot dulu sebentar untuk komunitas kami. Nah, jadi buat Bapak Ibu yang mau menyalakan video dipersilakan menunjukkan senyumnya untuk kita saling menyapa di sini. Nah, iya baik. Okay.
Okay. Maybe I will translate also in English. Oscar, is it okay? Okay. Now we are planning to take a photo and a screenshot for today's webinar, so we can probably turn on our video, and we will screen capture all the faces that show in the slide of the video. I mean, of the webinar, I'm sorry.
Yeah. Please show your best smiles. Oke, kita mulai dari slide pertama karena ini pesertanya antusiasnya lumayan tinggi ya. Jadi banyak sekali pesertanya sampai 4 slide. Jadi mohon ditahan senyumnya Bapak Ibu, rekan-rekan seluruh peserta. Mohon ditahan senyumnya. Kita memiliki 4 slide di sini untuk di screenshot. Bisa dibantu dari tim atau saya yang screenshot.
Okay, probably for the screenshot, it will take some seconds. So please hold your smiles as long as you can. And yeah, probably it can start from now.
Oke, ini untuk slide pertama. Sebentar, tahan. Okay, the next slide, the second slide. Please keep your smile. Oke, boleh dihidupkan kameranya yang belum menyalakan kamera. Oke, saya akan capture di sini. Oke, 1, 2 untuk yang kedua ya. Kedua. Oke, nah sekarang bergantian ke slide ketiga. Slide ketiga, mohon ditahan senyumnya karena kita nggak tahu Bapak Ibu, rekan-rekan ada di slide nomor berapa. Nah, mohon ditahan senyumnya dulu.
Yeah, please hold your smile as long as you can.
Slide ketiga. Oke, sekarang untuk slide terakhir.
And for the final slide.
Please. Alright. Ya, sudah semuanya tercapture di sini. Jadi nanti bisa kepoin langsung sosial media kita. Mungkin ada gambar Bapak Ibu di sana. Oke, terima kasih atas partisipasinya sekali lagi untuk Bapak Ibu dan rekan-rekan yang berpartisipasi di webinar hari ini. Once again, thank you Kelly Kim for presentations. Hi. Okay.
Okay.
Waduh, anak. Lagi ngidam kayaknya.